His work not only advanced our understanding of developmental and regenerative biology, but also hold exciting translational potential for cardiovascular medicine. His topic today is characterizing the epicardium during the zebrafish heart regeneration. Please join to welcome Dr. Wang here today.
Yeah, so nice introduction. So hello, good morning, everyone. So I'm really excited to be here today to share my work in the past several years. So I have been here. I have been in UGA for several times, but this is the first time I'm a speaker to be here. So okay. So today we are share about heart regeneration. So in zebrafish, you can see here. I don't know people you already see zebrafish before. So
Slide one.
okay. So we are interested in heart regeneration. The reason is the human heart, human heart couldn't regenerate cardiomyocytes after myocardial infarction. So, but we know after the heart disease, like the myocardial infarction. So if there's no blood supply to the cardiomyocytes, the cardiomyocytes will die and then replaced by the permanent scar tissue and without new cardiomyocyte production. So there's currently there are several methods to try to treat the heart failure patients, like the heart transplantation and also like the cell therapy, almost maybe some drugs, but all of these methods have its own problem. So currently there's no final solution for the heart failure treatment. So for many years, heart has been considered as a post-mitotic organ. So that means after birth, the cardiomyocytes couldn't proliferate. I mean, couldn't produce new cardiomyocytes. But the interesting thing is I think it's around in the past 20 years, we have many interesting discovery in the mammalian heart. People try to label the cardiomyocytes, and then they follow the labeling they found in the humans after birth. They can see around like 1.5% cell cardiomyocyte turnover annually. Even at around 75 years old, you also can get around 0.5% the turnover. So this means the human heart has the capacity to regenerate cardiomyocytes, but for some reasons they couldn't efficiently produce enough cardiomyocytes or complete this process. So we think if we can understand the mechanism of natural heart regeneration, we can help design the better strategies to do heart regeneration therapy. So the model my lab used is the adult zebrafish. So adult zebrafish, they will take around 3 months to be mature. I mean, they can produce the next generation. So the fish is around 2 to 3 centimeters. So zebrafish have a 2-chamber heart. It's different from the humans, because humans have 4 chambers, but they have a similar function. Like the atrium, they can save blood from the whole body, from the body, and then the ventricle will push the blood out of the heart. So here's the outflow tract. But if we check the cardiac tissues, we found they are very similar. They are the same cardiomyocyte tissue. Like the epicardium cells is the mesothelial layer. I'm on the outer surface of the heart. So in the layer is this labeled green one is the endocardial layer. They can line in the inner surface of the heart. And in the between is the myocardium. This is the major contract tissue. So, so also this myocardium can be divided to two different populations. From the histology is the close to the ventricular wall. This is the compact muscle, and here is the trabecular muscle layer. So we are actually seeing is to understand whether this myocardial cell can proliferate after heart injury. So this we know this from 20 years ago. So it's this is actually here is the Camposis, my previous postdoc mentor. So during his postdoc, he found that after this is the cartoon we show if in the heart we can cut the ventricle apex up to 15 to 20%. This heart will quickly form the blood clot, and then they can regenerate like normal within 30 to 60 days. Here's the section of the heart. You can see there's the uninjured heart. And if we cut the ventricle apex here, then we get the 30 to 60 fully recovery of the injured area. They include the lost cardiomyocytes and the epicardial cells, myocardial cells, also the corneal vessels. They can fully recover. And also there's no scar formation during after regeneration. So in this past 20 years, so zebrafish regeneration field is very dramatic, exciting discoveries. We found that, so first we found that vertebrate heart can fully regenerate. So before this recovery, before this discovery, people don't think that vertebrate heart can regenerate. So that's very important, the concept change. Another thing we found the cardiomyocyte source. So after we observe this phenomenon of heart regeneration, so we would think it's a stem cell or progenitor cell contribute. But finally, we demonstrate this cell source is from the existing cardiomyocytes, and the mature cardiomyocytes to differentiate, and then give to new cardiomyocytes. Also identify the cardiac tissues to heart regeneration, like the epicardial cells, endocardial cells, and the immune cells contribute to this process. We also found the oxygen effects on cardiomyocyte proliferation. And then if there's a high oxygen level, the cardiomyocyte proliferation is very low. But if there's no oxygen effect, the cardiomyocyte proliferation can be increased. So we also identified the regeneration elements, like some DNA sequence. I mean, they can induce the regeneration response. Also during this process, during this period, we identified many different molecules involved in this process. Some can promote this cardiomyocyte proliferation, but some maybe may inhibitory effect. So this is the regeneration of some different time points. You can see here is the after the heart surgery, around like several hours. Within several hours, you can see that we have formed blood clot here, and then we can see the epicardial cell. As I talked, the epicardial cell is the cellular element of the whole heart surface. And after the injury stimulation, the epicardial cell very quickly expand, and then they will gradually migrate to the injured area. And and finally they will during this 3 to 7 days this period. There are many signals will increase or some molecule decrease, and then they make the environment to let the cardiomyocyte proliferation very quickly, and then they will produce enough new cardiomyocytes to restore the lost cardiac tissue. So currently there are many cell types have been studied, like the epicardial cells, cardiomyocyte, and cellular cell, corneal and cellular cell. Also immune cells, like the neutrophils and the macrophages, and also T cell, Treg cells. But today we will talk about epicardial cell function during this process. Also, we are mentioning some macrophage regulation mechanism. So epicardial cells, here you can see we use the epicardium markers, the T-cell21 regulatory sequence to generate a back transgenic reporter. This is using the nuclear GFP, can label the epicardial cell nuclei. So you can see this is the the epicardium cell, they can even distribute in the heart surface. So after the myocardial ablation, so we use the genetic method to express the differential terpsin A in cardiomyocytes, and after ablation, and up to 60%, the heart can regenerate, and fully regenerate. We also can observe the very robust epicardium response. Within several days, they can become very dense. So when we observe this phenomenon, we think at that time point we don't know the cardiomyocytes. The new cardiomyocytes derived from the existing cardiomyocytes. So people are saying that maybe some stem cells or progenitor cells can contribute to cardiomyocytes, but finally we found it's not. But another possibility is that people think this epicardium cell maybe also can produce, can transfer to change to cardiomyocytes. So to test this possibility, so we use the T-cell21 proposed to drive the Korea, and then with the GATA4 switch line to do energy tracing. So so one is that we check the development process. So we enable treat the fish, the double transgenic fish with the 400 tamoxifen in around several days post fertilization. And then we suppose now we can enable the epicardium cell with the nuclear GFP. And then we wait the fish to grow to adult. Then we check the heart, and then we didn't find any nuclear GFP positive cardiomyocytes, indicating that epicardium cell couldn't contribute to the cardiomyocytes during development. So another possibility is the epicardium cell might contribute to cardiomyocytes during regeneration. So in this time we treat the fish with the tamoxifen at the adult, and then we do resection surgery. And then wait 30 days, and then we check the heart injury area. This time we also didn't see GFP positive cardiomyocytes, so indicates the epicardium couldn't contribute to new cardiomyocytes. So the next question is we want to know the epicardium cell function, because they can robustly respond, but they must play something. So to do this, we first question we ask is whether epicardium cell is required for this heart regeneration process. To do this, we use a strategy use the neutral reductase system. So in this system, this is the enzyme. They can transform the prodrug beta-nitrol to an active form, and when this MTZ is activated, they will make the DNA cross link, and then the cell will die. So we use the T-cell21 transgenic the promoter to drive the neutral reductase, this enzyme, in the epicardium cells. So when we give the MTZ treatment, the MTZ will be activated in the epicardium cell, and then they will kill the epicardium cell. So we can get around up to 95% epicardium cell ablation. This is the hallmark heart. You can see very clearly the decrease of epicardium cell thickness. So in this phase, we first treat the MTZ and then ablate epicardium cell, and then we perform the resection surgery. Wait for another 30 days. So we stain with the myosin heavy chain staining to indicate the myofibers. You can clearly see that in the control phase, they can regenerate the cardiomyocytes, the north path. But the epicardium cell ablated part, they show very obvious gap in the injury area, indicating the cardiomyocyte restoration is blocked if without the epicardium cell. So next, we confirm this process is affecting cardiomyocyte restoration by staining the PCMF2 staining. So PCMA is a marker for reaching cells. MF2 can enable the cardiomyocyte nuclei. So you can see after if we in this phase, if we ablate epicardium cells, the cardiomyocyte proliferation is significantly dropped. So that means the epicardium cell is required for myocardial regeneration. So now we know that, but the thing is that for many years we already know the epicardium cell is a mixture of different cell clusters. So at that time point we think that epicardium cell different type of population maybe do different things, but we have no evidence to do that evidence. So we performed the single cell sequence of the epicardium cell. So first is the uninjured heart. We can find that is the 7 clusters that have different function, but then we compare with the regeneration heart, we find that's also similar cluster number, but that show many different response, like the cluster 1. They can increase from dramatically increase the number, and the number 2 or the other mainly is decreased. So the function analysis indicate that like the cluster 1 has the major mainly correlated with regeneration process, like the ECM deposition, organization, and also muscle development. So we think this one may be very interesting population. Then we check the gene expression in our single cell sequence results. We found haploid 1A is the can immediately express in the cluster 1. But actually, we found is if you want to find some specific gene expression in your cluster, it's difficult. So we do check several one, but this one is the best one. And then we use the back transgenic to generate haploid 1A GLP reporter. So we can observe the EGLP signals is located in the ventricular wall in our site. And here is the high magnification. You see this cell is lying in this compact layer. So we cross this line with the epicardium cell reporter. You can see this GLP signals is co-localized with the arbitrary signal indicating the haploid 1A cell are T cell type 1 positive cells. But you can see also some arbitrary signals here. But there's no GLP expression indicating this haploid 1A positive cell actually is a part of T cell type 1 positive cells. So next, we also examine the haploid 1A expression in the myocardium and also the endocardium. So from these reporters, we found there's no overlapping red and GFP signals. It indicate their haploid 1A is not expressed in these tissues and very specific in the epicardium cell. So next we will examine whether the haploid 1A can respond to the heart regeneration or their behavior during this process. So we found in the injured area, the uninjured heart, there's also mechanism mainly in the surface of the heart. But after heart surgery, it's the 7 days post amputation. You see the epicardium, this haploid 1A positive cells is localized around the injured area, and then they accumulated in the injured area during this process. And the 30 days, we also observed some haploid 1A positive cell localized here. So we want to know whether this haploid 1A positive cell correlated with the cardiomyocyte regeneration. So first thing we used this GATA4 GLP reporter. So GATA4 GLP reporter has been used to demonstrate to enable the proliferation cardiomyocytes. Because they are at 14 days, we observe they are in a very similar area. So we check the double transgenic phase. We found the GATA4, this GLP positive proliferation cardiomyocytes is lining with the haploid 1A, the GLP positive cell, this cell. So that means they are very closely interacting with each other. So we back to the development process because from the regeneration study we know the many regeneration events actually recapitulates the development process. So we first check the juvenile heart. So during this in zebrafish juvenile stage, there is a myocardium compaction process. Means there's many cardiomyocyte production during this period. So people already use this GATA4 reporter. They can also use GATA4 to enable the preliminary cardiomyocytes. If on the cardiomyocytes will appear in the ventricular base, and then these cardiomyocytes will expand and then form the compact layer. So we checked the happening on HLP report. We found it doing around the 5 weeks post-fertilization. This age is around this the GATA4 positive cardiomyocyte appear. So we found there's very few happening positive cells. But in later time points, the happening this GLP positive signal will expand, and then they will gradually interact with each other and recover the other surface. So we check this double transgenic phase during this process. We found this happening positive cells actually is very close to the GATA4 positive cell in the similar area. So in this higher magnification, you can see this GATA4 positive cardiomyocytes actually is very perfectly linked by the happening positive cells. So here's the whole heart image. So we section the heart. We also can see the happening positive cell is lining the proliferation cardiomyocytes. So next, we check whether this happening positive cell is required for myocardium production during this compaction process. So we treat we use the gamma photopia cell with the haploid NTR phase. When we treat the phase with the MTZ, and we found that after we ablate the haploid positive cells, the GATA4 cardiomyocytes actually is decreased, significantly decreased. And then we examined the heart. We found after the 30 days post this ablation, the heart actually looks similar, a little smaller. But if we check the myocardium, is the compact muscle layer, we found that there is much less compact muscle layer in this if without the haploid positive cells. So next, we check whether this haploid positive cell are required for myocardium regeneration. So we use this, you can see on the 14 days. If we ablate the haploid positive cell, the GATA4 signals, GATA4 prolific cardiomyocytes is dramatically decreased. Also, we check the 30 days with the check the 30 days and the myocardium recovery. So we found this without the haploid positive cells, the myocardium regeneration is totally blocked. So next, we want to know what's the molecular mechanism during this process. So one interesting candidate is this happening one gene itself. So here happening 1A is a ECM protein. They can binding and stabilizing this hyaluronan proteoglycan complexes. So mutant mice have died shortly after birth. So there's no really study in the adults about the happening one expressing the function. But we to understand the haploid function. So we generate this haploid 1A mutation. I mean, firstly, luckily we get this haploid 1A mutant, homologous mutant is available. And unlucky thing is when we check the 30 days post mutation, we found the heart actually haploid 1A heart can fully regenerate. So looks like we don't know what don't know. Looks like haploid really resolves during this process. And then we checked whether there's some compensation effect happened here. So we checked this happening 1A homologous happening 1B. And we found this happening 1B expressed in the epicardium cells. To confirm the expression, we generated the happening 1B, the arbitrary reporter. So we compare with the happening 1A GLP, we can get more than 95% co-localization of this two signals, indicating they actually exactly they express in the same cells. When there are also some very few cells they don't really overlapping. The one reason, I think we guess the reason is because the virus transgenic reporter they can repeat the replicating the endogenous expression, but for some reason they couldn't label the 100% of the cells. So we couldn't get the 100% colonization, but this really many crossing each other. So we generate the happening 1B mutants. And in the homologous phase, we found the myocardium regeneration actually is blocked in this phase. So we actually try to compare the happening 1 and the happening 1B sequence. But the current we don't know what's the reason. Of course, they are different functions, even they have very similar protein on the structure. So next we want to know what's the downstream target of the happening-1 gene. So the one thing is the hyaluronate acid, because the happening-1 can regulate the hyaluronate and protein-glycan complex stability, and they are binding. So we think maybe that's the downstream, maybe we regulate the happening this HA structure. So we check this has one is the hyaluronate synthesis enzyme. So this enzyme is the major enzyme for the HA production. So we check the HAS1 expression. We found that HAS1 expression is expressed in the class 1. So we suppose this. So we guess this happening-1 is expressed in the same cell as the happening-1A and the happening-1B. So we check this happening-1A GFP with the HA staining, antibody staining. We found happening-1A positive in the injury area. This happening-1A positive cell actually is very close to HA signals, indicating the happening-1A positive cell may regulate the HA structure. So we also check the HA signals with the GATA4 prolific cardiomyocytes. We found the GATA4 positive the signal cardiomyocytes actually is lining with the HA outside. So why we interested really check the HA structure or signals with the happening-1 or cardiomyocytes because in the bioengineering study, there are many they generate the matrix for the in vitro cardiomyocyte proliferation. This HA is like the HA is the main component actually in this matrix cell, but people do not know the this HA function in vivo cardiomyocyte proliferation. So I think our study provide evidence that HA can really regulate the GATA4 protein cardiomyocyte proliferation. So next we examined these cells during in junior fish heart during the myocardial compaction. We also observed that this happening-1 positive cells is tightly closed with the HA deposition. And then this HA cells also, I mean, you can see the HA signal is also lining outside of the GATA4 positive cell. So indicating this happening-1A positive cell and also the HA deposition. But since the deposition is tightly correlated with the cardiomyocyte proliferation. So next we want to check. We check whether the HA is really affected by depleting happening-1 positive cell or knock out the happening-1 gene. So in the junior fish heart, we can see this HA structure is lining a lining structure in the ventricular wall. But after the depletion, we saw this structure is this lining structure is disrupted. Also, we from the quantification, we found HA signals also is decreased, indicating the HA production is reduced after depleting happening-1 positive cell. So in the injury heart, we can see also this is HA structure in the injury area. It's also like some lining structure. But after the depletion of happening-1 positive cells, you can see this is more likely from the aggregates of the HA signals. So this will indicating the happening-1 positive cell is required for the HA production and also their organized structure. I think this one may be very important for the cardiomyocyte proliferation. So next we also confirm that this one is in we examine the happening-1A or 1B inactivation effect on the HA deposition. So we found that after look out the happening-1B gene, we found that HA signals actually we found there's no change, but their structure is decreased. That's the disorganized. So we think this outside is a 3D ECM component is disrupted, so the cardiomyocyte couldn't proliferate. So after we know the happening 1 and 1B is critical for the zebrafish heart regeneration. We want to know whether this also play a role in the mammalian heart. If we check the mouse heart, we can see in the fetus in the heart there are many HAPLin-1 positive cells, but in the adult or even after the heart injury, the myocardial infarction, there is very few HAPLin-1 positive cell response. So here is in the human heart, also in the fetus heart, there is many HAPLin-1 positive cell. But in the adult and after the heart failure, in the heart failure patients, there's a very few HAPLin-1 positive cell. I mean, there. So from this comparison, we think the HAPLin-1 maybe is very critical for cardiomyocyte preservation. So in the regenerating hearts, there are many or there's environment for the cardiomyocyte preservation, like in the embryonic hearts, they must have many HAPLin-1 positive cell. But the later in the adult heart or after the myocardial infarction, for some reason, the HAPLin-1 expression couldn't be induced, so the heart couldn't regenerate. Currently, we don't really verify this speculation. I mean, hypothesis in mammalian heart. The summary here is the first part is that we know the epicardium cell subpopulation can surround the cardiomyocyte proliferation area, and also the HAPLin-1 positive cell and the HAPLin-1B are required for new cardiomyocyte proliferation. So HAPLin-1 cells, we think the HAPLin-1 cells mainly regulate this process by producing like the ECM components or regulates the ECM structure in the heart. So next thing is that we want to know what's the whether they have other functions. So we compare with the HAPLin-1A and the 1B negative cell. And then we found that HAPLin-1A positive cell has actually enriched with the angiogenesis factor. So we think maybe HAPLin-1A can regulate the corneal vessel growth. So this time we use reporter is the we identified several years ago is the delta CGOP. So this line can indicate the corneal vessel on the surface. So previously we use the anosmia cell marker like the FLI1 or FLI1 reporter to label the anosmia cell, including the vessel cells. But there's always background. So for this line, we can very clearly only just see the corneal vessel. So in this double transgenic phase, if we ablate the HAPLin-1 positive cell, we found after the 30 days this post ablation in the regenerated heart, we found there's a very few corneal vessel formation here, indicating the HAPLin-1 cell may affect the corneal vessel formation. Also, we checked in the juvenile fish heart during the cardiac compaction process. We found after the depletion, the HAPLin-1 positive cell, the corneal vessel cell, the structure actually is disrupted. Also, the whole amount of the corneal vessel is also reduced. So we think so kind of we don't know what's the reason. So we checked the HAPLin-1 positive cell and the corneal vessel in the juvenile fish heart. So at the next stage, there are many corneal vessel growth in the heart surface. So we found that this HAPLin-1 positive cell actually is surrounding the corneal vessels. So I think this one can explain why we observed the disrupted corneal vessel in the heart surface, because the HAPLin-1 positive cell can play a role like the perivascular cell. So perivascular cell can stabilize the newly formed corneal vessels. So if we deplete the HAPLin-1 positive cell, the corneal vessel will be disrupted and form like the disconnected cells, the vessels. But the interesting thing is we also observed there HAPLin-1 positive this structure looks like there are also some tube there, but there's no vessel there. We also found some cell like corneal vessel tips. there's no haploid positive cell. So that's what we decided to examine what's the main type or what's the ratio of them. So we calibrate this phenomenon with several different populations. And one is the coronary vessel tips, even they separated were ahead of this haploid structure. When they are in the same area, it will always keep the same. Or is the haploid positive cell form some structure is ahead of this coronary tips. So after statistical quantification, we found actually the major phenomenon, major thing happened is the haploid positive cell actually can ahead of coronary vessels. So that's very interesting for us because vessel guidance, vessel patterning is growth pattern. I mean, is a very hard. Is unknown. We don't know how this pattern. So we think maybe this haploid positive cell can guide coronary vessel growth. But to test this possibility, we used. Okay, so sorry. I mean, we also checked this phenomenon in the injured heart. So this is the 14 days post heart amputation. In this wounded area, we check this delta this indicating this coronary vessel tips. We found the always located area is with the haploid positive cell, and from here this 3D this image you can see this looks like this coronary tips also behind this located in this haploid positive cell area. So indicating also this haploid positive cell may also guide coronary vessel growth. So to test this possibility, we use the ex vivo system, it's heart ex vivo system. So we can take the juvenile fish heart or adult or zebrafish heart and then culture in the dish. And so in this medium, we second this incubator, and then we can observe the heart daily in this under microscope. Actually, we can count this heart for several weeks. Heart also can keep their morphology and also keep beating. So we can use this system to monitor like the cell behavior, like the epicardium cell, coronary vessel cells in the heart surface. So we first culture the juvenile fish heart. We also use this reporter. You can see you can see this area. You can see just only 2 or 3 days. We see a lot of coronary vessel growth in this heart surface. Also, we culture the adult heart with the heart injury. You can see here is the day one. You can see a very big chunk of the there's no coronary vessels. But at around 3 days, we got lots of coronary vessel here, from new one coronary vessel here. So currently we don't know why this coronary vessel grows so fast compared with the in vivo system, because this process always happen maybe several weeks. This process was always happening in around 1 or 2 weeks. But in our system, they can just in several days. So we think one possibility is this media maybe contain some paragraph factors. They can stimulate this regeneration, this coronary growth process. But anyway, we can use this system to examine their interaction with the epicardium cell. So to do this, we take the juvenile fish heart out, and then we found some area like this coronary vessels, but also there's haploid positive cell area. So you can see here, okay, we are going to here after the 6 hours. After 6 hours, we found this delta C DNA positive cell actually is grow, extend a lot. So if we compare with this haploid positive cell, we found that this haploid positive cell already pre-existent there. So that means this haploid can follow this coronary can follow haploid positive cell structure. And after 12 hours, you can see more coronary vessel growth in this area. At the beginning, there was also a haploid positive cell there. So that indicates this haploid cell might can guide coronary vessel growth. But you also can see some coronary vessel is indicated here. So 6 hours. So there are some coronary vessel this cell growth happened here. But here there's no haploid positive cell. But after 12 hours, we found this area, this coronary vessel actually is withdrawn. So that means this haploid positive cell not only just guide the coronary vessel growth, but also is required for maintain the coronary vessel extension. So next question we want to ask: so what's the molecules they can play? They can play a role in guidance the coronary vessel growth. So we check the many vascular angiogenesis factor and angiogenic factors, and also some factors affect the cell migration. We identify this SEPAN1. Means is very interesting because this SEPAN1 is a factor that can affect the neuron migration. So in the uninjured phase heart, there's no SEPAN1 expression. But after heart surgery, the 7 days in the injured area is always in the coronary vessel growth happen area. We found this SEPAN1 is expressed in this haploid positive cells. So you can see from here, this looks like this delta C is indicating the coronary vessel tips actually is located in the area with the SEPAN1 expression. So we next examine whether this SEPAN1 function is required for coronary growth. So we don't have the genetic tools to specifically block the knock out SEPAN1 or block this pathway, but we use the drug. It's the nectin. So that is used for blocking this SEPAN1 function. So we found in the juvenile phase heart after the drug treatment, the coronary vessel growth is significantly blocked. Also in the regenerating heart, we found coronary vessel growth in the injured area also blocked. So indicating the SEPAN1 function is required for the coronary vessel growth during heart morphogenesis and regeneration. So summary 2 is the second part is we know the haploid epicardium cell depletion can block coronary vessel growth, and also the epicardium cell can guide coronary vessel growth, and the SEPAN1 which that is the critical point. Current molecule can regulation is guided guide the coronary growth. So I will spend several time also another time to introduce about the another epicardium cell function. So already talk about the haploid expression in label is class 1 function, but we also interested to see is about the immune response. So after the heart injury in mammals or in zebrafish, the heart injury can induce our robust the inflammation response, and then the neutrophil will come very quickly, and also macrophage will come. And now we know the macrophage have several different populations, phenotypes, like the inflammation phenotype, also have the reparative phenotype. So how we don't know how this macrophage be correlated to stimulate heart regeneration. So this question we don't know the answer. So we are interested in whether the epicardium cell can play roles for the immune cell response. We found this class 2, like class 2 have the function can regulate, can involve maybe immune response. So we also do the cell chat analysis to compare to examine the zebrafish heart or also the neonatal mouse heart, the macrophage interactions with other cells. We found actually there is a very tight communication between the epicardium cell or fibroblast with the macrophage. So why we use this fibroblast or epicardium cell, because the zebrafish, both the mice hearts, the main is the fibroblast, but the fibroblast is derived from the epicardium cell. But currently we don't have a very good marker to really separate the fibroblast and epicardium cells. So we combine them together to do the single cell sequence analysis. We found you can see here is the macrophages in zebrafish have the strongest or more interaction with fibroblast and macrophage epicardium cell. Also in the neonatal mouse heart, we also can observe the stronger, the most tight collection with the macrophage and also the epicardium and the fibroblast. So we checked the gene expression. So next first we confirm this interaction with the reporter. So we use this T-cell21 to enable the epicardium cell. Also the GLP reporter to enable the macrophages. So we found this epicardium cell very always localized. I mean the macrophage always localized with the epicardium cell in the similar area. If we use the T-cell21 NTR phage to deplete epicardium cell, we found there's very few macrophage in the injured area during the regeneration process. So these results indicate the epicardium cell they associate the macrophage, but also the require rate for the macrophage recruitment during regeneration. So next we checked we want to label this subpopulation is this class 2. So we check the gene expression in the single cell sequence results. We found the PTS3A gene is expressed in the class 1. It's transient upregulated after the heart injury. So we generated the PTS3 GLP reporter. We found in the injured heart there's no PTS3 expression, but after heart injury, we found that PTS3 expression is upregulated in the injury area, in the natural injury site, and then these cells we are accumulate in the injury area, and then through several weeks, but then we disappear after the regeneration done. So the next we also use a similar strategy, and we use the generating the T-cell20 PTS3 NTR phage to ablate the PTS3 positive cells. So after this cell depletion, we found this also observed the macrophage number is decreased in the injury area. Also we observed that in this heart, even there's no PTS3 positive cells, there are many collagen deposited in here. So indicate the inflammation response is increased. So next we want to know because the MPT-GLP reporter can label what macrophages, including the inflammation macrophage, also include the reparative macrophage. So next thing we want to know what's the phenotype, what's the macrophage phenotype correlated with the PTS3 positive cell. So first we also confirm this PTS3 expression cell with the MPT, the macrophage cell, and also you can see that what is look at together close to. Then we use this CSCR4B antibody staining. So this is a marker for reparative macrophage. So we can find that this cells, this CSCR4B signals only very close or surrounded by the PTS3 positive cell. So indicating the PTS3 positive cell actually is mainly localized to the CSCR4B positive cell. And then we checked with the TNF, we confirmed this one with the TNF alpha GLP reporter. So this line can indicate the inflammation or fibrotic macrophage. You can see there's very few this TNF alpha positive cells in this TPS3 area. So we quantified these signals like the CSL4B, TNF alpha, these signals with the PS3 M3 signals distance. The MRC1 1B is the marker also can enable the reparative macrophage. So after the after qualification, we found this mainly is this reparative macrophage with the PS3 positive cell, and TNF alpha cell is very few. So we think this PS3 can selectively associate with reparative macrophage. So next we confirm if we deplete the PTS3 positive cell, we found there is very few CSL4B signals in the injured area. If we check this TNF alpha GLP positive macrophage, we found there is not very big difference between them. Even with we observed a little little increase. We think because the reason is because the PS3 cell maybe might interact with the leukocytes and can transform the TNF alpha positive cell to CSF4B cell. But if there is no PS3A positive cell, there should be a little more increase of TNF alpha positive cell. But now we're working on this. So next, we want to know what's the really molecules can play and derived from PS3 positive cell. So we also analyze the zebrafish heart, also the neonatal mice heart. We found that among them, one is one specific one is the CSF1 is pathway. So CSF1 alpha is a macrophage cloning stimulation factor, and it can promote the macrophage functional survival. But there's no study about very few study about that function during the heart repair and regeneration. We don't know which cell can secrete the CSF1 alpha. So we from this single cell sequence results analysis, we found PS3 actually express CSF1 alpha. So we did the single did the fish the fluorescence in situ hybridization. We found the CSF1 alpha is expressed in PS3 positive cells. I mean, cells you can see from the expression. Yeah. Here, see. But fish the fluorescence in situ hybridization. I mean, sometimes the signal is very hard to detect. So that's the reason we got a little many. Currently, we are generating the CSF alpha report. So in future, we want to directly see how the cells overlapping or not. So next, we checked whether CSF1 alpha is important for the macrophage recruitment. So we generated the CSF1 alpha mutant. So in the CSF1 alpha homologous mutant, we found the CSF4B positive macrophage numbers decreased. So indicating the CSF1 alpha is required for the positive macrophage recruitment. So next, we checked the collagen deposition in the area, and we found that after the block out the CSF1 alpha gene and block the reparative macrophage, that's increased collagen deposition. So the inflammation response increased. So next, we also ask whether if we increase the CSF1 alpha, we can get more reparative macrophage to benefit the heart regeneration. So we use this Cranox P16. We can express the CSF1 alpha in the injured heart. So we will observe more macrophage actually in the injured area. And in this fish heart, we also detect more CSF4B positive macrophage, indicating the CSF1 alpha can increase the beneficial macrophage for the heart regeneration. So during this period, we observed the less TNF alpha GLP cell macrophage in this area, indicating this CSF4B this CSF1 alpha overexpression can transfer the nuclear site, the progenitor cell, to the reparative macrophage. So we also examined the PS3 and also CSF1 expression in the regenerating animals, neonatal mice heart, compared with the P8 is non-regeneration neonatal mice heart, and we found that at the 1 day post injury in the P1 mice heart, there's a much more PS3 expression and the CSF1 expression. So currently we're trying to collaborate with Mass Lab. You can check this PS3 and the CSF1 effect on the macrophage recruitment and also on the heart regeneration. So summary, the third part is we know the PS3 positive cells associated with reparative macrophage. And if we deplete the PS3 positive cells, we will reduce the reparative macrophage recruitment and also increase the scar deposition in the wound. If we manipulate the CSF1 alpha, we can block the macrophage recruitment if I get O expression, we can increase the heart reparative macrophage response, and then finally they can benefit the heart regeneration. So this work today we're talking about work is mainly performed by first postdoctor decision, and it's here. And our work is supported by the Emory startup from the division of the cardiology, and also grants from the NIH and the American Heart Association. And I will stop here to take questions. Thank you so much for your time.
Thank you for the great talk. I'm curious whether the heparin 1 also expressed in other cells in addition to epicardial cells.
Yeah, in the heart, there's in the ventricle, there's only expressed in the epicardial cells. There's not expressed in other cells. We confirmed with the reporter. Yeah.
How about the immune cell? Other immune cell?
Immune cell, there's no heparin expression in immune cell.
Thank you.
Yeah.
Yes.
So I noticed that although in mouse and human hearts, the heart count is very low,
Yeah.
and after injury, they still increase. So do you have any?
Which one is that? Yes, it was or.
Actually, we are in one.
Oh, yes, yeah, yes. Okay, I can show you why that's different.
Besides showing mouse and human.
Yes, that's this one, right?
So after heart failure in human heart, we see an increase.
Right.
But what could be the function of this?
Right. So actually now we think you can say if we compare with the fetal heart, that's right, or in mice or the high human, you can even there's some increase, but the number actually compared with this one is very few. That's right. But of course there's more compared with the healthy heart. But we think in the mammalian heart, the adult heart is because they couldn't enough. Maybe this heart is not enough, so we couldn't really stimulate many mouse that proliferation can really efficiently regenerate. But also we know that even in the diseased mouse heart and human heart, we also can detect some mouse that proliferation happen there. But just the number is very few. So we think even there's some increase, but it's not enough. But if we increase more hepatitis positive cell, we get a more strong response. Have mouse and proliferation. Yeah, right. Yes.
So this positive cell, probably 1A,
Yes.
for the revascular is very amazing. So you think about the comments as more structural guidance, or they need to be there to secrete some kind of what we call morphogen or something to guide the formation of these tubes.
Yes, actually we don't. Yeah, I think we don't know too much about that. So now we are generating the many reporters or workouts. We try to identify maybe some molecules can guide or this growth. But I think because the hepatitis positive will already form a structure, genetic structure in ahead of the coronary vessels. So the one possibility is they can build like some road, that's right, can pave the road. So maybe there's no guidance effect. But because now already road, so the coronary vessel can very easily grow in this road. Another possibility is they can secrete some guidance effect. They can just attract them to growth in this way. So currently we don't know which way is this main reason. But I think at least the ECM component in this area form the structure is very critical.
Yeah, just think on the therapy side, should we really transplant that cell in a way, or it doesn't matter, we just need to inject it there, then they may secrete something to guide it, or we have to put it in the right place.
Right. Actually, when I listened to the bioengineering talk in the industry, they try to use some the tube. They use the ITA or some cell, the ECM there, and then the vascular cell can grow it can form the tube. So I think so. Currently, we're doing things we try to separate this hepatitis positive cell to different populations. One maybe some population of hepatitis positive cell can only surrounding the prolific cardiomyocytes, or some some haploid positive cell, this cluster can only localize the head of the corneal vessel tips. So if we can identify this cell population, and then in future we can specifically identify the molecules in cells.
So you think it's kind of like because for the features, right? You see this positive cell or even the PTX3 cell, they are maybe more abundant. So you want is more like a rejuvenate or promote regeneration.
But yes, we want to get a more specific one, and also we can really isolate them one, or can differentiate them to this specific cell population, and then we do cell therapy. I think we can get a more efficient treatment effect. Yeah.
Yeah, I think so today is 12:30. Thanks everyone, and thanks Dr. Wang again.